next speaker is Gisela Storz, who will talk about, I think, something quite appropriate. The small things matter. Gisela has a BA in biochemistry from the University of Colorado at Boulder, PhD in biochem from UC Berkeley. She worked with Bruce Ames, so that, I think, tells us a little bit about what she might be talking about. And I noticed you postdoc with Sankar Adia at NIH, good friend. Fellow of the American Academy of Microbiology, American Academy of Arts and Sciences, the National Academy, and received the ASM Eli Lilly Award. So terrific. Gisela?
Thank you. Well, I thought I would tell a little bit about the research being conducted in my lab. I'm at the National Institutes of Health right now, and at the same time reflect a little bit on what made a difference in my career, what little things or maybe bigger things made a difference, and I also want to end with what I think are still some challenges. And the theme is the small things. And in part that comes from the focus of the research in my lab right now are small regulatory RNAs as well as very small proteins. And I mean proteins of less than 50 amino acids. So these gene products are ones that have been missed. They have been ignored. So people don't think about proteins that are so small. And for many years, people didn't appreciate that RNAs are regulators. This lists some of the reasons why these genes were missed. They were not annotated. When genomes are sequenced, the genes are identified, and these very small genes were missed. They're missed in genetic screens, so you can't easily get mutations in them, in part because the genes are small, but also because people ignored them. They're missed in biochemical assays. When people analyze proteins, often they use techniques where the small proteins are just run off the gel. So I've been interested in these small RNAs and small proteins, and I actually started thinking about the small RNAs as a graduate student. And I've been lucky throughout my career. These are the people I've worked for. You can see all my mentors, their last names started with A, Albersheim, Ames, Adia, and Asabel. I don't know how that happened. And I was always in cities that started in B, Boulder, Berkeley, Bethesda, and Boston. But I was lucky. All of my mentors, they didn't play a very active role in what I did, but they just let me do what I wanted to do. They sort of stood by and with sort of amusement, whatever, you work on this project. And so I started working on regulatory RNAs back in the mid-'80s because I did the wrong experiment in graduate school. I made a mistake. This is what's called a northern blot, where I was looking at RNA, and I was planning to look at the band at the top, and then I used the wrong gel and I used the wrong probe, and I saw this small, tiny transcript here. And it was just so striking because the expression was induced by a stress condition, by hydrogen peroxide, that I decided to figure out what was going on. And I did this northern blot when I was a second-year graduate student, and I finally published it 12 years later. So, but I had mentors who allowed me to keep playing with it. But this discovery of this RNA, which we realize is acting as a regulator to base pair with messages to affect their stability and translation, made me become interested in how many other RNA regulators are out there. There have been a number of different approaches, serendipity, computational approaches, direct detection, which have allowed the detection or the discovery of many more, not just in bacteria, which I have focused on, but now in eukaryotes, it's really clear that regulatory RNAs are key in a lot of regulatory systems. I just want to summarize some of the general concepts we've learned, both in bacteria and in eukaryotic cells. Many RNA regulators act by limited base pairing with mRNA targets, so they can base pair and they can affect translation or they can affect the stability of the target. But there are also RNA regulators. In bacteria, this base pairing oftentimes requires a helper protein, a chaperone protein, which in E. coli is called HFQ. But there's also RNA regulators. So the focus so far has been on the RNA regulators that act by base pairing, but there's also a number of examples in bacteria of RNA regulators that bind to proteins, and I think the same is becoming more and more evident in eukaryotes as well, that RNAs can also act as regulators by titrating proteins. These are two examples in bacteria. This one RNA, the 6S RNA, is one of my favorite RNAs. This was characterized by a postdoctoral fellow in my lab. The reason it's one of my favorite RNAs is that the sequence was published in 1971. It was one of the first nucleic acids whose sequence was reported, and people didn't know what it's doing. And what Karen Wasserman in my group found is that it binds to RNA polymerase, which is central to the transcription of all genes. And so a question was, why had no one found this RNA associated with RNA polymerase? Well, everyone had always used the same protocol to purify RNA polymerase. Hundreds of labs have purified RNA polymerase. No one had ever seen this RNA. That's because everyone used the same protocol, which was designed to remove nucleic acids. So the protocol was designed to get rid of this small RNA, which we found acts as a regulator. So I just want to summarize that these RNA regulators in bacteria and other organisms are key to many different regulatory responses. In bacteria, they are induced in response to a whole variety of different stress conditions, limited iron availability, oxidative stress, when you transition from an aerobic to an anaerobic environment, and they are part of many complex regulatory networks, which is in part summarized here. So you can have these small RNA regulators where multiple small RNA regulators act on the same target. You can have them in negative feedback loops. So they're really key parts of a number of regulatory networks. So my lab has spent many years now studying these regulatory RNAs, which has been great fun. But we've now also started this project to look for these small proteins and study small proteins. And how did we come into this? As I said, they were ignored. People didn't annotate or document proteins that were less than 50 amino acids. Well, this in part came because we stumbled across these small proteins in our studies of the small RNAs. So in many cases, the small RNA regulators are called non-coding RNAs. In other words, they're thought not to code proteins. They're non-coding. And I specifically don't use that term because I think in many cases it hasn't been clearly documented that in fact these small RNAs don't encode small proteins. And I say that because we've stumbled across a number of small proteins in our studies of small RNAs. And one example is here. This wasn't what we thought was a regulatory RNA. We called it RIB-A for an RNA regulator, but we didn't know what it was doing. But then when we mapped the start of transcription, the +1, and looked at the sequence and the conservation, we saw that the conservation was indicative of a protein. So the conservation of a nucleic acid sequence for structure is different than the conservation of a protein. And this had conservation of a protein, 2 nucleotides, and then what's called a wobble base, 2 nucleotides. So we realized that this small RNA actually encodes a small protein. And this made us start to look for more proteins, more small proteins systematically. Again, we've used mainly computational approaches to look for conservation that's indicative of conservation of a small protein. But what was important in these studies is that we then documented that these small proteins are in fact synthesized by adding a tag to the gene and showing that we could detect the small protein. What I did not appreciate when we started this work, and so this is a documentation that we can detect these small proteins. What I did not appreciate when we started this work is that the majority are in the membrane. I didn't expect this, but over 50% have what's called a single transmembrane domain and are in the membrane. And I now think that this is going to be a major way to regulate membrane proteins. So I just want to mention one example that we found and then just point out that in the literature there are other examples. But we have shown that the ones we expect to be in the membrane are found in the membrane when we fractionate the cells. So they not only are predicted to be in the membrane, they are found in the membrane. But one of the ones we've been studying, we now have given the name ACRZ. It's pretty large. It's actually 49 amino acids. And since we have these small proteins tagged, we can purify them and pull them out. And when we did this, we could see two proteins that co-purify with ACRZ. And one of these was ACRB. And we found this very interesting because this is one of the major drug efflux pumps in E. coli. This is one of the reasons E. coli cells are resistant to so many antibiotics. And this pump had been characterized quite a bit. ACRB is in the inner membrane of E. coli, and it's connected to Tol-C, which is in the outer membrane, by another protein called ACRA. And this complex of proteins has been studied quite a bit. It's been crystallized. The structure is known, but no one had seen this small protein. And what we think this small protein is doing is affecting the ability of this pump to pump out certain kinds of antibiotics. So cells that have mutations in ACRB or lack this blue component of the pump are sensitive to a whole variety of antibiotics shown here. This is a competition assay where we mix wild type and mutant cells, and the mutants are sensitive to all of these different antibiotics. Those cells that lack the small protein are also sensitive, but only to a subset of these antibiotics. So we think the interaction of the small protein with the pump and somehow affects the ability of the pump to export certain classes of antibiotics. And that's just illustrated here. So normally this ACRB, ACRA, Tol-C pump can pump out drug A and B. Here it's more efficient with drug A. But in the presence of this small protein, this tiny protein interacting with the larger ACRB protein, now it's also effective at pumping out ACRB. So we're at the very beginning of these studies. I'm really excited because now we have structures of the ACRB together with the small protein, and one can imagine how maybe one could use very tiny proteins as drugs or ways to modulate membrane proteins. And I say this in particular because now other groups have also discovered tiny proteins affecting kinases that are in the membrane, which is the case for a protein called MGRB, and proteins that affect cell division. So you can imagine how these small proteins interact with the bigger proteins to affect their activity. And as I said, I'm going to predict that this is going to be a bigger theme. So that's sort of where my research is at the moment. But I thought I'd use this opportunity to reflect on the things that have made a difference in my career. Sometimes I think that's useful because hopefully then I can help students or postdoctoral fellows that are in my lab. I think I was very lucky when I was a graduate student. I was a graduate student in Berkeley in the mid-'80s, that for some reason the cohort of women that were in graduate school at the same time I was incredibly strong. And we were very supportive, but also a little bit competitive with each other. I would say that's still the case, but it's really helped all of us sort of egg each other on or be supportive of each other. So several are professors, there's the dean, associate vice chancellor. One of my roommates went on to win the Nobel Prize. So it really was quite a supportive environment. I think in my life that made a very big difference. I have 3 children, and my husband is also a successful scientist. I think I've been very lucky that my family has been quite tolerant, but I would say it's not always been easy. I also have benefited, and I would say many of these themes have come up before, but I think they do bear reiterating. I have had some very supportive colleagues that have made a huge difference at different points in my career by providing the kind of encouragement that was mentioned at lunch, that said, you're doing a good job, you can do it, when experiments didn't work. I would say another thing that has been really critical to me has been female faculty friends. So I found that as graduate students, you have a cohort. As postdoctoral fellows, you have a cohort. But sometimes when you're a faculty member, a female faculty member, you don't have as many friends, and you have to act very positive, and things are going well, and yes, the grant was just rejected, but you are going to try again. And so I found it very useful to have faculty friends, or especially female faculty friends, who are going through the same and could be supportive. And one group is a group, and you'll notice they're all women, with whom I've written a molecular biology textbook. So many women in my neighborhood, the moms, have book clubs where they get together and discuss books, but clearly not books about scientists, fictional books about scientists, because they don't sell. But this is my book club. We get together and we write a molecular biology textbook. But it's been great fun because not only do we talk about the textbook, but we also complain about kids and having teenage kids and that sort of thing. So that has been really important. I have found, and this is something I didn't expect, I have found incredible joy in mentoring. That I thought what I really wanted to do was just to do research and to do experiments. But I would say, looking back at the over 20 years I've been at NIH now, where I've really gained satisfaction and joy has been in mentoring and watching the people from my lab go out and have successful careers and go in different directions. Not always something that I would have expected, but I definitely embrace. So to me, it's been great fun. I feel that's a great privilege that we have in this profession. But as enthusiastic and as lucky as I feel in my career, I think it's also important, as has already been done in this forum, to talk about the challenges, and I think there are still challenges. I have labeled them as small challenges. Maybe they're not so small, but I think it is important to talk about them. I would say, as supportive as my family is, it's not easy to have 3 teenage kids and to be trying to run a lab and to, go to meetings and give presentations, and to know when to turn down an invitation to give a presentation. And I thought the article by Anne Marie Slaughter was really important in articulating this. This problem hasn't gone away. My kids are very active. They haven't been interested in science, but they've been very competitive athletes in various arenas, which as a parent does take time. And how much time do you devote to supporting your children versus mentoring someone in your lab, listening to a talk, or giving a presentation at a conference? I also am still, I would say, disappointed that the leadership in many places hasn't changed more than it has. This was from a C-SPAN presentation on December 6th. And while I really appreciate that all of these institute directors went and commented on how important research is for the country and why it is important to support funding for NIH, this is still the public image of science in many ways. So these are several of the very prominent directors at NIH. And I would say, since I've been at NIH for over 20 years, I'm a little bit disappointed that this hasn't changed more. And I think another thing that does need to be talked about still is the reduced funding. That I worry disproportionately affects female scientists. An example of this, so with limited funding, NIH comes up with all kinds of new initiatives and new bench to bedside or pioneer awards. And I sometimes worry, as these awards are implemented, that it's a little bit of an old boys network, a little bit, how the awards are presented. An example is the Pioneer Awards, where the first year no women were awarded a Pioneer Award. And I think that continues to be a big challenge, especially with 6% funding cutoffs for grants, that this needs to be discussed. And I'm very glad that it's been mentioned how scientists need to educate the public and continue to be proactive in saying how important science is, because the funding is important. But I don't want to end on a negative note. Like I said, I feel like I've been incredibly fortunate. And I want to just bring these topics up for discussion with the hope that the people who are currently in my lab, which is the people on the left, and people who have been in my lab, and many of whom are running their own research programs in their own labs, will have the opportunities that I have and will be able to experience the excitement for as many years that I have. So let me end there. Thank you very much. Yeah.
I noticed this. Oh,
Go ahead.
What do you think will change the climate at NIH in terms of the old boys network? I mean, do you see that changing?
You know, I'm a little bit discouraged, and I don't know exactly how it should be changed. So there's a lot of lip service to things are going to change. We care about this. And there's task forces, and I was disappointed that the outcome of the task force, which I think is important, is to get more child care, of which there still isn't enough. So I don't know why this hasn't changed more. And that's why I feel like now I'm going to start speaking out. As that was said here, I'm going to be a little bit more sort of in people's face about why is this not changed.
Well, let me just say that if we can help you in any way, we certainly will, because we do get the word out, and that's one of the mandates of the society. And we'll do it, and we'll do it even more vocally. And if we know you'll be by our side, we'll call on you to do it. Francis
he talks a good talk. He does. He's very good. And I was here when he presented.
Then we'll have to hold him to it. Do you think there should be term limits for these positions?
I think that's an interesting question. Some of it, there is some benefit to people who have institutional memory.
Oh, yes.
So that's the disadvantage of term limits. I feel that more active commitment to making sure the leadership is diversified is where I think that we should start. But I know in my institute, the director has changed twice since I've been there. And there was a woman candidate, but then they didn't choose her. Same for the scientific directors. I mean, part of it, there is a more limited pool. I acknowledge that in people, I think, but I don't think that's all.
Well, they wouldn't have to leave. They could become emeritus.
But I think this should be discussed.
I noticed positively, not a criticism, that those who have left and completed work with you have gone on to a variety of employment, which is very interesting because the expectation in the past has been that everybody needs to a merit praise go to an academic institution, but things are changing. Can you comment on that?
Yeah, and I would say I've definitely changed my view. I feel my role now is to have people as happy as possible in their job and their career. I mean, I feel like doing research isn't easy, and you have to love it, especially if you have a family, you have to love it. But so I really strongly try to work together with people to find what career they think they will love. And I sort of have changed my mind. I feel like we need to be training politicians. I really think scientists, I'm trying to encourage my postdocs now, go run for office and get into Congress, because we don't have scientifically trained people who are politicians.
Could raise the IQ a little bit too.
I think that's a perfect point. When we talk with my daughter and my youngest daughter, and we talk about science career, like my wife and myself, we did fundamental science, we change, we move to applied science, we do business, technology transfer. We always talk about Margaret Thatcher. We talk about Angela Merkel. I mean, both were scientists. Angela Merkel, Prime Minister of Germany, was a quantum chemist. And Margaret Thatcher graduated from chemistry as well. So you can start, and that's what I encourage young girls to do, thinking that science is only a tool, and a tool that you can apply for science, you can apply for everything. How many mathematicians we have in Congress? How many chemists?
They can't even count, never mind the math.
So it's nothing wrong to start a career in science and apply the science to serve the people through politics, through government, because that's what people do not understand. It's only a tool, and I think we need probably more scientists in Congress.
I really think, especially at the last government shutdown, there's sort of more women and more scientists in Congress.
Yeah, okay, Samantha, you know what's
Well, you notice that it was the women who actually got Congress
Yes.
Right.
back in action.
Came together,
Right.
cross-party lines.
Any other questions? We'll go on to the next speaker. Thank you very much.
We can do it 5